My name is Zhiyong Wang, and it's my pleasure to introduce Julia did her PhD in plant molecular biology with Klaus Schwarzheimer at Technical University of Munich, and she moved on to do postdoc with Bernhard Kuster in proteomics. I think she kind of recruited Bernhard into plant proteomics, and currently she's leading a newly established proteomic center at the hospital campus of the Technical University of Munich. So Julia is an early career stage scientist, and she actually just became the leader of the new proteome facility last year. She has done a great job in plant proteomics, generating the first comprehensive tissue level proteome atlas in Arabidopsis. So today her talk is on protein expression profiling in Arabidopsis tissues. So Julia, please go ahead, take over.
Thank you very much for this kind introduction. So let me just share my screen. Right. So I'm very happy to be here in the conference and about this opportunity to present some of the aspects of the Arabidopsis study that I did as a postdoc in the lab of Bernhard Kuster at the Technical University of Munich. So as Zhixiang already told, today I will talk about protein expression profiling in Arabidopsis tissues and how we can use this kind of large-scale resource data sets to investigate some existing biological hypotheses and also to generate new hypotheses. So we all know that starting from the genome, we have then a large variety of different transcriptomes and even a larger variety of different proteins, modified and unmodified. And these proteins can then interact in very different combinations, which even increases the complexity even more. This high variability of the proteome enables an organism that using the same genome information, it can produce a variety of different cell types and also react with different growth morphology to different environmental conditions, for example. So this is important, especially also for plants, which can react with very different growth morphologies to different, for example, light conditions. So if you grow a seedling under light, you get the phenotype with nice green cotyledons. If you grow a seedling in the dark, you get a very long hypocotyl because the plant tries to reach the light. Now, we already heard two great talks about different aspects of spatial proteomics. So when I actually hear the term spatial proteomics, I always think about subcellular proteomics. So where you have a specific localization of your proteins in subcellular compartments, but of course you also have specific proteomes in different cell types, which actually enable the cell types to perform their specific functions. And then on the larger scale, you also have different spatial compositions of proteomes to actually generate different organ types or tissue types of the plant or any organisms that you are studying. So our project actually dealt with this level. So we were interested in comparing different Arabidopsis tissue and organ types to generate a global overview about their specific proteome compositions. So for this, we selected 30 different sections of plants and also included some tissues grown in cell culture. And with our selection, we cover all of the major organ types of Arabidopsis plants. So for example, the leaves, the root system, different stem sections, different parts of the flower, the pollen, the fruit, seed and seeds. And we also have two different developmental stages in our atlas. So we have tissues from adult plants as well as from the seedling stage. With our project, we wanted to answer the questions, which of the genes can we actually detect as proteins? Where in the plant can we detect them? And also in what quantity can we find them? So our proteomic workflow looked like this. So we had our different tissue sample types, and we performed standard protein lysis and tryptic digestion. Then on the peptide level, we performed an offline fractionation step using H-sex chromatography and fractionated each sample into 24 fractions, and then measured all of the samples on a Q-exactive HF mass spectrometer in data-dependent mode with a 2-hour gradient. And all of the data analysis was then done in MaxQuant. So here in the middle, you can see with the bar chart, so these are the number of proteins that were identified for each of the tissues. On overall, we identified more than 12,000 12,000 proteins for each tissue, and combined together, this adds up to 8,210 individual proteins that were identified, and this is about 65% of the annotated protein coding genes in Arabidopsis. All of these proteins also had very different intensities. So here you can see they span several orders of magnitude in our study. And here, for example, we had Brevix radix like 3 identified on average as the protein with the lowest intensity, and the large subunit of Rubisco was identified as the protein with the highest intensity. So this picture here is an average over all of the tissues. However, if you look at the individual tissues separately, you can see a very different dynamic range in different tissue types. So here in these plots, we have on the x-axis always plotted the different proteins ranked according to their abundance. So number 1 would be the top 1 most abundant protein, the number 10 would be the 10th most abundant protein, and so on. So we have here for the seed sample, the 3 most abundant proteins are the cruciferine 1, 2, and 3. And you can see that already these 3 proteins make up about more than 20% of the total intensity that we measure in this sample. So here on the y-axis is plotted the abundance increase for each protein, and in total this makes up to 100% for all of the proteins in one sample. So we have this very specific dynamic distribution for the seed sample. However, in most of the other samples, this looks more evenly distributed. For the leaf sample, for example, we have typically the Rubisco large subunit identified as the most abundant protein. Then here, the second most abundant protein is another subunit of the Rubisco complex, and we have the Rubisco activase as the third most abundant protein. And together, these 3 proteins make up less than 10% of the total intensity in a leaf sample. All of the other tissue samples showed even more evenly distributed intensity distribution across all of the proteins. You can also see this in this plot over here. So we also looked up what was the top 1 most abundant protein in each of the tissues. And when we did this, we came up with a list of 11 proteins in total. So they are listed here. And then we plotted here what was the proportional intensity of these proteins in all of the different tissues. So you can see here that for the seed samples, which are depicted in brown here, we have a very high intensity of cruciferine 1 again. And for all of the green tissues, so leaf tissues as well as stem sections, we have between 2 and 4% of intensity from the large subunit of Rubisco again. What you can also see here is that although these proteins like Rubisco or cruciferine are very typical for specific tissues, that doesn't mean that they only appear in these tissue types, but we can also find them in most of the other tissues actually. This is something that we also saw when we were looking for tissue markers actually. So we found only very little proteins which are truly only detected in one particular tissue. So for a large number of proteins, however, we found an enhanced expression in a particular tissue type as compared to all of the other tissues. The largest number of these kind of tissue-enhanced proteins we actually found for the pollen sample. So the tissue-enhanced proteins are here shown in dark blue. So in pollen, we found about 1,200 proteins, which were, as you can see here in this heat map, very high expressed in pollen compared to all of the other tissues. We also find a high expression of these proteins in the stamen sample, which is the closest similar tissue to pollen in our atlas. And we also find a little bit of expression in the other flower samples. Now, when we do gene ontology enrichment of these proteins, we actually end up with the terms pollen tip growth, cell tip growth, developmental cell growth, and for cellular compartment, we get an assignment for pollen tube or the endomembrane system. So this fits very well to the results that we have, that it is that there are pollen proteins, and this nicely confirms our analysis. So we can also use this type of tissue expression clustering to also gain information about proteins for which there's currently either very little or no annotation actually. So here we selected a number of proteins which are uncharacterized and hypothetical currently, for which we have a very nice tissue-specific expression pattern. So here in the top, the first protein actually has a typical leaf expression pattern, what we call it. So we have a high expression in all of the green tissues. And this protein was actually already found in a chloroplast protein study in 2006. So this all, again, confirms our study very nicely. For the second protein, we see a similar trend. So we have here very high abundance in the pollen. So we would say this is a protein which has a function in pollen. And we also found the annotation in tear actually that this was already assigned to the pollen in the pollen proteome study in 2005. Here the protein, this is assigned as a seed proteome protein sample. And we also find information that this was actually already reported as seed localized on transcriptome level. However, now we also can add the proteome level information. But we also have cases where the annotation does not agree. So for the last example down here we have actually also an annotation in tear as chloroplast localized, so like the first protein, but based on our results, we would actually rather say this has a function in cell division. So we can use co-expression to gain information about a specific function of proteins, so to specific compartments, but we can also use co-expression to find interaction partners. So we wanted to test if our dataset actually has sufficient resolution to do this. So we used an dataset of Arabidopsis protein expression, the reported protein expression dataset, where we have a lot of pairwise interaction partners, and we calculated the pairwise Pearson correlation of these protein interaction partners in our dataset. So when we do this, we get here a distribution of the Pearson correlation of all the protein pairs. And you can see here in blue, this is the distribution of the Pearson correlation coefficients on protein level, and gray is depicted on transcript level. So if we use the whole data set, we actually get a quite low average Pearson correlation of just 0.23%. When we split the data set based on the method that was used to identify the protein interaction, we get for protein interactions that were identified by affinity purification MS a slightly better average Pearson correlation coefficient. However, the coefficient is still quite low in these results. So what we marked here in blue actually is the percentage of interaction partners where we would say this is protein action partners where we believe we could find this based on our data set, and we come up with this cutoff actually based on a study which we did with known complex subunits. So we selected a couple of very well known and annotated complexes and calculated the pairwise Pearson correlation of all of their subunits. And then based off the Pearson correlation that we detected for these complexes, so here is just depicted a couple of examples, we then decided that 0.5 is our lower limit that we would expect for true complex partners as a correlation in our dataset. So here, for example, we have the chaperone complex, which has a very, very high correlation between all the subunits. On the other hand, we have the Kopnine-Signalosome complex, which has an average Pearson correlation between the subunits of just roughly 0.6. So our conclusion from this analysis actually was that our data does not have sufficient resolution to identify interaction partners without any additional information. However, if we look at already well-established and known complexes, there's a lot of information that we can actually gain from the tissue atlas. So here we are looking at the coatomere complex, which is a cytosolic complex that is required for budding from Golgi membranes and retrograde Golgi ER transport. It is composed of 7 different subunits. And in this case, actually, several of the subunits are encoded by paralog genes in the Arabidopsis genome. Now, for mutant analysis, it is often quite important to know which of the paralog form is the dominant one, because if you want to knock out specific gene and see what its function is, if you have a redundant paralog which can take over the function, you might not see a phenotype. So we were interested to see which of the paralogs actually are the dominant form, or if they are both present in equal levels. So here we, on the right, we compared the proportional abundance of the different paralog forms of the different subunits. And we can see here, for example, for the subunit alpha, we have mostly a more dominant form for the second subunit. For the paralog form 2. But in some tissue samples, for example, for the flower, we have a more abundant alpha 1 subunit. For the beta subunits, we actually have a more even distribution of both paralogs. And for the beta dash paralog, we have actually 3 different forms, and they are roughly in equal abundances across all tissues. Now for the epsilon subunit, we can clearly see that the epsilon 1 subunit is a dominant form, and epsilon 2 has barely any abundance at all. So probably epsilon 2 has no real function anymore. The zeta subunits are quite interesting because here we see that for the zeta 3 subunit, we have a dominant abundance in most of the tissue, except for the seed samples. So here in the seed samples, we have actually a zeta 1 as a dominant form. So we can not only have different paralogs, we can also have different gene families which actually provide a variable subunit to a complex and thereby change its function. So here we are looking at the exocyst complex. So the exocyst complex is a plasma membrane localized complex which is functioning as a tethering complex of Golgi vesicles to the plasma membrane. It is composed of 8 different subunits, and we have a core complex of the 6 SEC subunits, which actually are quite stable in abundance across all of the different tissues. And we have 2 variable subunits, so EXO84 and EXO70. If we look at EXO84, so this a subunit exists in 3 variants, A, B, and C. And we can see that in most of the tissues, EXO84A is actually the dominant subunit. However, for the pollen sample, we can see that here EXO84B is the more important subunit. For the EXO70 subunit, this is actually more complicated, but this is a family of 24 different proteins, and we could identify 16 of those in our proteome study. So we can see here that these different proteins exist in varying abundance in the different tissues, but for most of the tissue, we have comparable pattern, so just with slight variation. So here, for example, for the root and for the leaf, we have little bit changing amount of subunits, but in general, the pattern looks similar. However, again, for the pollen, we have a very different picture. So here, actually, the subunit C2 and C1 are the dominant forms. So this might be related with the fact that in pollen, you have a very active physical transport system going. And actually, it has been reported already that the C2 and C1 exo70 are required for pollen tip growth. So going even one step further, so you can not only have different subunits, but you can also have different versions of a complex which act in different cellular compartments. So here is an example of the chaperonine complex. So the chaperonine complex is required for protein folding in Arabidopsis plants and also in other organisms. And here we have a version of the complex which is located in the cytoplasm, one version which is in the mitochondrion, and one is which is in the plasmid. The cytoplasm complex is actually composed of 8 subunits, and we already saw this earlier in the Pearson correlation analysis. So here, the CCT1 to 8 subunits actually have a very, very nice correlation across our tissues. And we can also see here that the Pearson correlation among the subunits is very high. For mitochondrion complex, so here we have 3 different proteins which make up this complex. So here we also have a very nice correlation of the subunits with each other. For the plastid complex, the picture was a bit different. So here we actually have a very nice correlation for 4 of the subunits, which actually also show the pattern that we would expect for a plastid localized complex. So that they have a high expression in the leaf samples and a lower expression in the roots. Now, 2 of these complex subunits actually show different expression patterns. So you can see here in red and in gray, these 2 proteins have actually a lower expression in the leaf as compared to the root. So this was a bit unexpected and led us to speculate that maybe these 2 subunits have a different function and act independently of the other subunits of the CPN60 complex in plastids. So to investigate this a bit more in detail, we made use of a different data set that we generated, where we used size exclusion chromatography to separate protein complexes according to their size. So here we used 3 different tissue samples, so flowers, leaves, and roots, and performed the native protein lysis and extraction, separated the complex over superosis X, 6 columns into 8 fractions, and then did a standard triptych digestion, and measured all of the samples again on the QX35HF system in DDA mode. And we analyzed the data with MaxQuant and also used the CC profiler algorithm to assign co-eluting peptides to their respective proteins. And in the next step, we can then assign the co-eluting proteins to complexes. So this data set gives us a very nice overview about the complexes that we can detect in the different tissues. And here we just selected as an example 5 complexes which very nicely span the size range, so between 100 and 900 kilodalton. And you can see here, these are the normalized intensity profiles for all of the subunits, and you can see that the subunits of each complex nicely co-elute with each other, and you can also see that the different tissues give very reproducible patterns for at least this selection of complexes. So there are also complexes which give a tissue-specific pattern, but in this case we selected the bonds which are very stable across all of the tissues. So we have also the intensity information actually of the subunits, which we can use to also assign stoichiometry information about the complexes. So here, for example, for the proteasome complex, we could see that we have a double elution peak of the core subunits. So we have here the very high peak for the core subunits alone. And then we have a second peak for the core subunits combined with the lid components, which are depicted here in light blue. Now, when we look now at the chaperonean complex, so we again could very nicely identify all of the proteins from the cytoplasma, from the mitochondrion, and also from the plastid complex. And what we could see, so the cytoplasma complex is composed of 8 subunits, so it is larger and elutes earlier than the mitochondrion and the plastid complex. Now, what we also saw is that the 2 interesting subunits of the plastid complex, so CPN60A2 and B3, actually co-eluted together with the other subunits of the plastid complex. So this means that they're actually still part of the plastid complex. However, what we could see is when we looked at the ratios between the different samples, so when we compared the abundance information that we had for the different subunits in flower, in leaf, and in root, we could see that for the plastid complex, we had in all cases the highest intensity for A1 subunit. But in the case of the root, we had actually a very high abundance also for the B3 subunit as compared to the flower and to the leaf tissue. So this led us to the conclusion that the B3 subunit is probably important in non-photosynthetic plastids, which are also present in root. And this might explain why we had this different intensity distribution in our tissue map. So for the alpha-2 subunit, we are still not completely sure what is the explanation here. So we could not see a striking intensity in root here, but this might also be possible that it's just part of a different chloroplast, of a different plastid type, and this is the reason why the intensity profile does not match the typical chloroplast profile that we would expect in the tissue map. So with this, I'm actually already at the end. So I hope I could show you that tissue profiling is a quite nice technique to characterize different tissues and to also classify tissue markers. You can use it to assign functions to protein based on their expression pattern in the different tissues. And it can also be useful to investigate protein complexes based on their correlation and composition in different tissues. So all of our data is available in the public repositories, and you can also access the atlas via the website Proteomics2B, where you can look up the expression in different tissues, and you can also access all of the spectral data and information for each protein. So with this, I would like to thank all of my colleagues and my group. So especially, of course, Bernhard Küster, who made this project actually possible, Martin Freino for bioinformatic support, and Matthias Wilhelm and Dr. Samaras for getting the Arabidopsis proteome into Proteomics2B. I would like to thank all of our collaborators from the SFB 924 for the help with plant work and getting Bernhard to do plant proteomics. I would like to thank the DFG for funding, and of course, all of you for your attention. Thank you very much.
Let's turn on my mic. Thank you very much, Julia, for that wonderful talk. It's amazing how much we can learn from tissue maps. I think it's a great resource for the community. I think we have time for maybe 2 questions. The first question is for quantitative normalization, how was it done? Did you use a pooled universal control sample?
No, so we use the same protein, so it's all label-free data, and we use the same input amount, and then we performed a total sum normalization of the samples.
So the next question, for different composition of subunits across the tissues is very interesting. Did you check whether these different compositions lead to different functioning or any chance of activity?
Yes, that would be the next step to do. So unfortunately, we have not followed up on these results, maybe at some point in the future. So we are not so much of a plant biology lab, unfortunately. So we will have to do this with some collaboration partners.
Or for the community, whoever
yeah.
studying those proteins, knowing where they are, and maybe they can follow up. Thank you very much. So I think next we will take a break